So good morning and welcome to today's webinar on immunogenicity in cell and gene therapy beyond anti-drug antibody testing. My name is Débora Basílio Queiroz and I am a product manager for cell and gene therapy analytics, and I will be today's moderator. So I'm joined here today by our speakers from Miltenyi Biotech, starting with Dr. Mark Schuster, R&D group leader who focuses on enhancing analytics in immunology and assay development with a particular interest in T-cell immunogenicity assays, flow cytometry, and lab automation. Mark is followed by Dr. Anna Baranska, R&D group leader focused on cell separation and myeloid cell immunology with a particular interest in the development of advanced immunology workflows and research solutions for immune cell isolation, characterization, and functional analysis. Next, we have Dr. Laura Schlase, R&D associate director for clinical assay and reagent development, who focuses on developing bioanalytical methods for clinical trials with emphasis on immunogenicity assays. Unfortunately, Laura cannot join us here today, but kindly provided a recording of her slides. So filling in and also joining us today, we have Dr. Ben Novak, Director of Bioanalytical Services, who is an expert on biomarkers, bioanalytics, immunogenicity, and translational infrastructure. So the agenda for today consists of an introduction to immunogenicity drivers and assay strategies. This will be followed by examples of standardized primary cell isolation for immunogenicity testing during drug development. And finally, a real-world example from us here at Miltenyi Biotech, biomarker-based clinical monitoring and immunogenicity assay validation case. Please acknowledge the following disclaimers. And with that, I'm happy to hand over to our first speaker, Dr. Mark Schuster.
Hello from my side. My name is Mark Schuster, and in the first part of today's webinar, I will talk about immunogenicity drivers and assay strategy. So immunogenicity is defined as a substance's property to provoke an immune response against itself. So practically, it means if we take an example of a virus, for example, SARS-CoV-2, that virus can activate the immune system. And furthermore, it can induce an immune response which is targeted against itself. So in drug development, the most prominent example when immunogenicity is used by design are vaccines, because the intention of vaccines is to create immunoprotection and immunological memory. And staying with the example of SARS-CoV-2, there were plenty of vaccines developed during the pandemic, and all of them, or most of them, were developed to raise antibodies, mainly targeting the spike protein of SARS-CoV-2, because the spike protein was known to be an immunogenic structure, also with functional relevance. And these are cases where immunogenicity is part and a target of drug design. There's also what we call undesirable or unintended immunogenicity in drug development, and that is an adverse effect. So for example, structures like penicillin, viral carriers, foreign species, immunoglobulins, or chimeric antigen receptors have the potential to be immunogenic because they are partially or in combination of different protein subunits immunogenic, meaning they have the potential to create anti-drug antibodies or anti-drug T cells or certain kind of innate cell responses. And these can limit the effectiveness of a drug or even completely neutralize the effectiveness of a drug. So we distinguish cellular immunogenicity from humoral immunogenicity, and throughout the introduction, I will keep here the example of the CAR T cells. Cellular immunogenicity means that the immune system is developing a T cell which can target the CAR T cell and then kill through soluble or other factors the CAR T cell, usually via a process which we call antigen recognition, and thereby the antigen-specific T cell eliminates the CAR T cells. Another way is humoral immunogenicity. So at the end of that process, an antibody is created which can target the chimeric antigen receptor. And that also can cause neutralization of the CAR T cells. So in principle, cellular immunogenicity causes T cells which can target the CAR T cell, while humoral immunogenicity raises a specific antibody targeting the chimeric antigen receptor. So the potential consequences of anti-CAR immunogenicity can be quite severe. So for example, it can result in limited expansion of the CAR T cells after transfer into a patient, and also to shortened or to limited persistence of those CAR T cells in vivo. In an extreme example, when we are talking, for example, about repeated dosing, so CAR after CAR therapy, it's even possible that the presence of anti-CAR antibodies or anti-CAR T cells can completely eliminate the transferred drug product, thereby leading to resistance to repeated dosing. So when we look at structures, for example, antibodies, or here again, chimeric antigen receptors, we can see that the sequences usually can harbor regions where there's a higher probability for immunogenicity. So, for example, in a standard third-generation chimeric antigen receptor, the ectodomain of the CAR is in certain cases a murine scFv, which is then also containing a linker. And the linker is a foreign species, and the murine scFv also a foreign species sequence, meaning that the immune system recognizes those domains as non-self. In addition, the CARs contain so-called interdomain junctions. It means that the transmembrane domain, for example, the co-stimulatory domain or the CD3 zeta chain, are fused to each other, which is naturally not occurring, and at the junctions, so-called neoepitopes are generated. So sequences which do not naturally occur in human beings, and therefore they are potentially re-recognized by the immune system as non-self. All of these potential sites for immunogenicity combined can in fact cause immunogenicity, and in 7 out of 8 commercial CAR drugs used for treatment of hematological malignancies were reported with anti-CAR immunogenicity. And the FDA, for example, and EMA reacted to that by now requiring risk assessments for immunogenicity. This is also in the interest, for example, of pharma companies which are developing CARs or other kinds of protein drugs, because they can test relatively early for potential rejection of the drugs they are developing. So to give you a clinical example, how cellular and humoral immunogenicity testing is used to make a clinical decision. So this is taken from a Nature Medicine paper in 2025, where actual Miltenyi products were used. So here a patient was 2 times treated with CD19 CAR T cells. In the first round of the treatment, you can see here days post-injection. The blue line indicates the first route of injection. There was a nice expansion phase of the CAR T cells, followed by contraction phase, and then the establishment of a steady state of CAR T cells in this periphery. However, when a second dose of CD19 CAR T cells was necessary, there was barely any expansion of the cells and no establishment of a steady state. They were not detectable anymore. And that was correlated with the occurrence of immunogenicity. So in this patient, a high amount of cellular immunogenicity was found, so anti-CAR T cell, as well as humoral immunogenicity, so the presence of CAR detecting antibodies. So coming now to the assays to determine and to detect immunogenicity. So these assays mainly deal with adaptive immune responses, meaning we are looking here first of all at the interface of an antigen presenting cell and a T cell, and here at the interaction of the T cell receptor with MHCs, class 1 and class 2. So one first idea about the potential immunogenicity of a drug is to screen the sequence for potential epitopes. That can be done, for example, with in silico tools such as NetMHC-PAN, or also with in vitro epitope analyses. So from Anna Baranska, in the second part of the webinar, we will learn more about MHC-associated peptide proteomics, one assay how to determine in the wet lab the epitopes which are presented via MHC class 1 and class 2. As I said, the first level to make an idea or to develop an idea about how immunogenic a drug is to look at the epitope. The next level then is the analysis of the T cells. And here we can first of all detect cellular immunogenicity after stimulation. So what you usually do in these assays is that you provide potential epitopes derived from a drug to a mix of cells, and thereby the activation of anti-drug T cells can occur, and that activation is detected. And there are a variety of those assays available how to detect activated T cells. So one predominant assay, which I will show you later, is flow cytometry, so to detect activation markers following antigen-specific stimulation. Another very prominent tool are ELISpot analysis, where the count of cytokine-secreting cells is determined. And other assays are variants of DCT cell assays. So for example, to determine proliferation of antigen stimulation, or as well secreted cytokines. And the last level to detect immunogenicity are, of course, the humoral immunogenicity detection of antibodies, so secreted anti-drug antibodies. And here we can distinguish anti-drug antibody tests from neutralizing antibody tests, but also the detection of antigen-specific B cells. As I said, today's focus is not that much on the humoral immunogenicity, but more on the cellular immunogenicity assays. So the immunogenicity testing can be found throughout the entire drug development process. So from preclinical drug development to clinical trials and also into commercialization. So in very early preclinical drug development, when only the protein sequence is designed, or the variants of the protein sequence are designed, in silico screenings can be done. So the before mentioned in silico epitope analysis or screenings for similarity, so known epitopes which can cause immunogenicity. Once from these protein sequence, candidate molecules are identified, which then can give rise to lead molecules. The real wet lab assays can occur. So here we have in vitro prediction assays, the before mentioned cellular immunogenicity assays, so flow cytometry, ELISpot, DCT assays, and also the in vitro epitope binding assays to determine the amount of epitopes present in the candidate molecule. And with that information, for example, a candidate molecule can be de-immunized or optimized into a lead molecule. And once a drug enters the clinical trial phase and the commercialization, usually in ex vivo monitoring of patients treated with this drug, still the cellular immunogenicity assays can be used, but then also humoral immunogenicity assays become more and more apparent, because then you have a complete immune system of a patient, which can react also with the production of antibodies. So having now a closer look on cellular immunogenicity assays, and here I would like to give you a very generalized workflow on how anti-drug T cells are detected via antigen-specific stimulation. So usually these assays start either with isolated PBMCs or with any kind of blood product. And these PBMCs or the blood product are then used to in vitro stimulate the antigen-specific T cells. And then these antigen-specific T cells are in various forms of analytics quantified or visualized. So the principle of that in vitro stimulation with PBMCs and the blood product is the antigen-specific stimulation of the T cells. So in the PBMCs and the blood product, APCs as well as antigen-specific T cells are present, and it's possible to feed the antigen-presenting T cells with the antigen of interest. Then the antigen is processed within the cell, and fragments of this antigen are then loaded on the MHCs of the antigen presenting cell. And in case a T cell receptor which can recognize that peptide MHC complex is present, this T cell gets activated. And the activation can be detected via various methods. So the before mentioned flow cytometry can be used to detect everything, cytokines as well as activation markers on the surface of the cells, as well as, for example, certain changes in the expression pattern. ELISpots are mainly used to detect secreted cytokines, and then spots are detected. So for drug testing, in that context, the source of the antigen can be either the complete drug protein. When we are talking about cellular therapies, it's possible to use a cell lysate, and what we prefer at Miltenyi is the use of peptide pools, which we call peptivators. And those peptivators are peptide fragments which have a length of 15 amino acids, and they show an 11-mer overlap to each other. And with this 11-mer overlap, we can span and cover the entire sequence then of a protein-derived drug or of any antigen of interest. And in terms of chimeric antigen receptor immunogenicity, we have recently released 3 of those peptivators. So a peptivator which is specific for the ectodomain of the most common CD19 CARs, the FMC63-SCFV. And we have also released 2 complete CAR immunogenicity testing peptivators, one for a CD19 CAR construct and one for a BCMA CAR construct. So looking now into detail of one of the methods, so flow cytometry, which is a very versatile method to detect anti-drug T cells. So we have released here a standard workflow, which starts with isolated PBMCs or with complete whole blood. And we take those cells from a donor or a patient and transfer them into a 96-well plate. And then we treat the cells into these plates with our immunogenicity peptivators, plus Brevellin A for the last 2 hours of the 6-hour incubation time. Then we perform an optimized intracellular and extracellular staining with our Miltenyi antibodies before we are doing flow cytometric analysis. So, and here you can see one example of the detection of anti-CAR T cells. Here on the left-hand side, you have the negative control, so untreated cells. And here on the right-hand side, you have T cells which were treated with our CD19 CAR peptivator. And here you can see the occurrence of the antigen-specific T cells which reacted to stimulation with this peptivator peptide pool. So we can here, with this workflow, analyze a variety of different activation markers, so TNF-alpha, CD104, and interferon gamma. And we can also distinguish here different subpopulations of reacting T cells, so CD3 cells, CD8, and CD4 cells, for example. And we have released that workflow as in 3 different versions as kit. So we have an antigen-specific T-cell analysis kit for PBMCs, but also for whole blood, which we provide without any kind of stimulus. So we can select the stimulus ourselves, or customers can collect the stimulus by themselves. So this kit is open in terms of the stimulation. And we have also released a kit where we provide along the kit also the FMC63 scFv peptivator as part of it. So one way how we can boost the sensitivity of the assay is, of course, Miltenyi's proprietary magnetic enrichment, meaning we can include an enrichment step of the antigen-specific T cells prior to analytics. And we again take here advantage of the activation markers and the secreted cytokines. So Miltenyi released in the past a lot of kits how to sort activated T cells. So for example, via the CD154 microbead kits or the CD137 microbead kits, as well as certain secretion assays, for example, interleukin-2 and interferon gamma. And the effect of or the benefit of the isolation of the activated cells prior to analytics, you can see in that procedure, which we call rapid antigen-specific T-cell enrichment, the rapid ARTE protocol. So here you can see the amount of antigen-specific T cells on the top before enrichment. Here on the left-hand side, the sample simply without any stimulation. And here you can see no activation of the T cells. But as soon as we treat them here with model activators, BKV and VP1, you can see activation of T cells occurring. So for example, here in terms of interleukin-2, interferon gamma, TNF-alpha. So when we include here an enrichment step prior to analytics, we can massively increase the sensitivity of that method, whereby you can see here much more events of the activated T cells compared to the situation before enrichment. And this rapid ARTE protocol is one of the most sensitive methods to detect antigen-specific T cells in the field. And now we will learn much more about magnetic enrichment of cells from my colleague, Anna Baranska.
So good afternoon, everyone. My name is Anna Baranska, and today I would like to talk about preclinical immunogenicity assays using primary human immune cells. And specifically, I will focus on high quality primary cell isolation and its support in developing reliable assay results. This slide provides an overview of the main preclinical immunogenicity assays using primary human immune cells. And we start with PBMC assays that assess broad immune activation. ELISpot and FluoroSpot measure antigen-specific T cell responses. DC activation assays evaluate innate immune recognition. Next, DC T cell assays assess antigen presentation and T cell activation, and finally, MAPS supplements those approaches by identifying naturally presented HLA peptides. Because all of those assays rely on primary human donor material, biological variability is expected. Much of this variability is driven by HLA polymorphism, which influences peptide presentation and T cell recognition. To obtain representative assessment of immunogenicity risk, studies therefore often include 20 to 50 donors, depending on the assay and desired HLA coverage. With donor cohorts of this size, standardized PBMC isolation and handling become essential to minimize technical variability. The most widely used method for PBMC isolation is density gradient centrifugation. While it is very well established and routinely used in research labs, it has several inherent limitations that can introduce technical variability. So first, PBMC preparation may contain residual red blood cells, platelets, or granulocytes, which then can interfere with downstream functional assays. Second, the isolation process itself can reduce cell viability and alter cellular function. Third, the procedure itself is really labor intensive and time consuming, making standardization challenging. And finally, manual layer harvesting introduces operator to operator variability, which becomes particularly problematic when working with large donor cohorts required for immunogenicity studies. Overall, those factors can impact PBMC quality, assay performance, and ultimately the reproducibility of immunogenicity data. So the question is, how can we reduce isolation and reduce variability? Studies have reported here up to 60% differences in PBMC recovery between technicians performing the same isolation protocol. To address that, at Miltenyi, we have developed a straight from PBMC isolation kits, providing standardized PBMC preparation workflow that can be combined with the Automax Neo and Multimax 24 separators for automation. This enables standardized PBMC input for both preclinical and clinical immunogenicity assays. In addition to improving standardization, the straight-from workflow substantially reduces processing time. Depending on the starting material, PBSC can be separated in as little as 25 minutes from the whole blood and leukopak samples and around 40 minutes for Buffy-coat. If you compare that to the density gradient workflow, the time saved is significant. This is particularly useful for large donor studies and time-sensitive samples. Beyond workflow standardization, an important question is whether the isolation method itself affects the cell biology. Here on the left, straight from isolation kits achieved comparable or slightly higher purity than gradient centrifugation, and also the recovery was either comparable or higher for the straight from protocol. The more striking result is shown on the heat map. Monocytes isolated with density gradient centrifugation exhibit increased expression of immediate early inflammatory genes, including JUN, IL-1 beta, 6CL8, and TNF. This transcriptional signature reflects cellular stress and innate inflammatory activation. In contrast here, this inflammatory program is markedly reduced following straight from isolation. For immunogenicity assays, this is highly relevant. You want to measure the immune responses to the therapeutic candidate and not the activation introduced during sample preparation. So now, if we can generate high-quality PBMCs with minimal isolation-induced activation, the next step is obtaining the specific cell population required for downstream immunogenicity assays. One of the most important populations is the monocyte, which serves as the starting material for generating monocyte-derived dendritic cells used in DC activation, DC T cell, and MAPS assays. Here we offer several untouched and positive magnetic separation strategies with options for PBMCs and also various blood products. These approaches provide all high-quality monocytes for downstream assays, and like PBMC isolation, those workflows can be automated. Many preclinical immunogenicity assays require primary CD4 positive T cells, particularly DC T cell co-culture as well as ELISpot assays require those cells because CD4 T cell help is key driver of anti-drug antibody formation. These assays are widely used as surrogate assay for assessing other risk during preclinical drug development. Depending on the starting material, we offer here both separation strategies for positive and negative CD4 T cell isolation. And here especially, I would like to introduce and highlight our new CD4 microbeads ultra pure for efficient CD4 T cell isolation. CD8 T cell responses are also increasingly evaluated in CGT programs as they can affect the persistence and durability of cell and gene therapies. To support these assays, we offer a range of positive and negative CD8 T cell isolation solution, including also here the new CD8 microbeads ultra pure, which provide excellent cell separation performance and can be automated. So what we have discussed so far has been aimed at one goal: generating high quality primary cells in standardized and reproducible manner. Automation is an important part of that strategy. Cell isolation workflows can be automated using the Automax Neo and Multimax separator, helping ensure consistent sample processing across studies and sites. Looking ahead, Maxcelerate separators launching soon will provide an even higher level of automation for larger sample volumes and up to 12 samples per run. And here, if you are interested in a demo or would like to be a beta tester, please feel free to let us know and contact us after this presentation. For preclinical studies, the automation and reproducible sample processing supports scalable screening workflows, while in clinical research, it helps harmonize sample processing across sites and also time points. So with the cells prepared, the next question is how those cells can be used in immunogenicity assays. One of the most widely used approach is the C activation assay, which assesses the innate immunogenicity risk. Here, isolated monocytes are differentiated into modices over 5 to 7 days using premium grade IL-4 and GM-CSF that helps to ensure reproducible DC differentiation across donors and experiments. The resulting modices are then exposed to the therapeutic candidate to evaluate innate immune activation and DC maturation. Responses can be assessed by surface marker expression using, for example, MODC analysis, cocktail kit, cytokine secretion can be analyzed with MaxPlex, and gene expression analysis can also be done. This assay provides an early screen for innate immunogenicity and supports candidate selection for downstream DC T-cell assays. If you are interested in implementing this workflow, we have a path here. A MODC differentiation protocol is available in our application section at the Miltenyi website. Okay. Following DC activation, DC T-cell assay is used to evaluate adaptive immune responses and potential immunogenicity risk. After MODC generation, DCs can be loaded with therapeutic candidate or peptidator peptide pools, which can serve as both standardized antigens or assay controls. In parallel, autologous CD40 cells are isolated and cryopreserved in FCS-free cryobrew medium. Then antigen-loaded modices are co-cultured with CD4 T cells at defined ratios, and further on T-cell proliferation, activation, and also cytokine secretion can be assessed. As CD4 T-cell responses are key determinant of ADA formation, this assay is widely used as an in vitro surrogate for immunogenicity risk assessment. The final assay I would like to highlight is MAPS, which identifies peptides that are naturally processed and presented on HLA molecules. Following antigen loading of modices, HLA-bound peptides are then isolated and analyzed by LCMS, providing a direct view on the naturally presented peptide repertoire. This further on enables identification of immunogenic hotspots and also supports immunogenicity risk assessment and also the immunization strategies. And with that, I have covered the preclinical immunogenicity toolbox, including standardized primary cell isolation and DC differentiation. And now I would like to hand over to my colleague, Laura Schlagsa, who will discuss clinical immunogenicity risk assessment and how those considerations translate into the clinical setting.
Welcome to the clinical immunogenicity section of this webinar. So far, we have discussed the workflows and technologies that support cellular immunogenicity assessments. Now I'd like to show you how these approaches can be applied in a real-world clinical development setting. As Miltenyi Biotech continues to expand its own pipeline of clinical assets, we have the unique opportunity to not only provide enabling technologies, but also to implement and evaluate these workflows within our own clinical programs. With that context in mind, let's now look at how we approached immunogenicity risk assessment for one of our clinical development programs. As mentioned earlier, biotherapeutics can elicit unwanted immune responses. These responses might have safety implications, reduce treatment benefits, and in severe cases, contribute to the termination of a drug development program. To proactively address these risks, we perform an immunogenicity risk assessment, or IRA. The purpose of the IRA is to identify potential immunogenicity risk factors and establish appropriate mitigation and monitoring strategies throughout development. The process can broadly be divided into 3 steps. First, identification of potential risk factors, which may originate from the drug itself, the target patient's population, the treatment regimen, or manufacturing-related attributes such as process impurities and excipients. Secondly, evaluation of the likelihood and potential consequences of these risks on both safety and efficacy. And thirdly, assignment of an overall immunogenicity risk level. For those interested in deeper discussion of the process, I highly recommend the publication referenced on the slide. Importantly, the overall risk level should guide your de-risking activities throughout development. It also helps determine the type of immunogenicity assay required and the extent of assay validation needed to support your clinical program. Based on our immunogenicity risk assessment and interaction with health authorities, we concluded that cellular immunogenicity is safety relevant for this program and should therefore be assessed as secondary endpoint. Consequently, a fully validated assay is required. At this point, it is worth noting that there's currently no dedicated regulatory guidance for cellular immunogenicity assays. The closest available document is ICH M10, which addresses bioanalytical method validation and study sample analysis, but explicitly exclude immunogenicity assessments. Furthermore, because we anticipate relatively low levels of immunogenicity in clinical samples, we adopted a fit-for-purpose assay lifecycle strategy designed to maximize sensitivity and precision, particularly in the lower assay range. Our assay lifecycle begins with a design of experiment, or DOE approach. In this phase, we systematically evaluate how individual assay parameters affect sensitivity, precision, robustness using statistical modeling. This is followed by a confirmatory phase, where we assess biological variability across donors and finalize the assay SOP. Next, we perform a pre-validation phase to establish all required performance characteristics. Up to this point, our studies were conducted using the model antigen CMV PP65. Let me mention here that Peptivator peptide pools provide standardized antigen stimulation for overlapping peptide coverage, enabling robust activation of antigen-specific CD4 positive and CD8 positive T cells, and therefore supporting sensitive detection of cellular immune responses. In the subsequent phases, the validation is repeated using FMC63 peptide pool. Once all predefined acceptance criteria are met, the assay can be released for clinical sample analysis. This slide provides an overview of the complete clinical sample workflow. At the clinical site, whole blood is collected and PBMCs are isolated and cryopreserved on the same day. For PBMC isolation, we use the MaxPrep PBMC isolation kit, which enables rapid isolation of highly viable, untouched PBMCs suitable for downstream immune function assays. One major advantage of this workflow is improved interoperator consistency. This is particularly important because sample processing is often performed by laboratory personnel with varying levels of experience in sterile cell culture technique. Following isolation, PBMCs are cryopreserved using StemMark's CryoBrew, a serum-free cryopreservation medium that helps minimize unspecific cellular stimulation. At a later point, frozen PBMCs are shipped to our GCLP-compliant laboratory for analysis. There, the cells undergo thawing and resting, each step followed by viable cell counting using the MaxQuant analyzer in combination with 7-AAD staining. The analytical assay is an interferon gamma ELISpot assay. In brief, peptide-stimulated PBMCs are incubated overnight on plates coated with interferon gamma capture antibodies. The following day, the cells are removed and secreted interferon gamma is detected using a biotinylated detection antibody followed by streptavidin alkaline phosphatase. Finally, a chromogenic substrate is added, resulting in the formation of visible spots that can be quantified using an automated ELISpot reader. Let's now look at the optimization of the pre-analytical workflow, specifically the PBMC thawing and resting procedure. Obtaining sufficient numbers of viable cells for the analytical assay is critical. Therefore, we use a DOE-based approach to systematically optimize cell recovery and viability. Among the parameters investigated were serum concentration, media temperature during thawing, the number of wash steps, the use of GMP-grade nuclease, resting duration, and cell density during the resting phase. This figure is meant to illustrate one example of our data analysis. Of the 7 factors investigating, resting duration proved to be one of the most influential parameters. Reducing the resting phase from an overnight incubation to 2 hours significantly improved both cell recovery and viability. An additional benefit was operational efficacy, as the overall workflow was shortened from 3 to 2 days. We applied the same DOE philosophy to the analytical phase of the ELISpot assay. Essentially, every step of the manufacturer's protocol was evaluated, including plate pre-blocking, incubation times, mixing procedures, and handling of the kit reagents. Moving to the confirmatory phase, this graph shows results from 3 representative donors categorized as high, medium, and low responders. When comparing the optimized protocol with the original procedure, we obtained improved signal intensity without a corresponding increase in background. This improvement was particularly pronounced in the low responder donor population, where assay sensitivity is the most critical. Overall, the optimized workflow resulted in clear improvements in both signal-to-noise ratio and log delta values. To conclude the assay development phase, all DOE findings were confirmed using multiple donors to account for biological variability. In addition, we investigated critical assay reagents and finalized our reagent qualification strategy. The first example shown here is the ELISpot kit itself. Across several kit lots, responder status remained consistent for all tested donor samples, including low, medium, high, and non-responders. The second example highlights fetal cough serum, or FCS, which proved to be a highly influential reagent. As shown on the right, equivalency testing across 4 FCS batches revealed noticeable differences in both background signal and peptide-induced responses. The key takeaway is that FCS should be treated as critical reagent, and sufficient quantities of qualified batches shall be reserved to support validation activity in subsequent clinical testing. One of the more interesting observations during assay development involved our functional control. Over time, we notice a gradual decrease in the functional control response. Initially, one might assume that this reflected a decline in PBMC quality or viability. However, when we reviewed the associated metadata, cell viability remained stable throughout the same period. Further investigation revealed an unexpected root cause: the storage temperature of FCS aliquots. After adjusting the storage condition, the functional control response returned to a stable and reproducible level. The broader lesson here is to continuously monitor your positive controls throughout assay development, not only during validation and routine testing. Trending changes in control performance may help identify previously unrecognized sources of variability, including factors that might not be discussed in the literature. Let's now turn to the FMC63 peptide pools and the generation of key validation parameters. 3 parameters are particularly important for cellular immunogenicity assays: limit of detection, or LOD, confirmatory cut point, or CCP, and minimal response. To establish these parameters, treatment-naive donors are assessed using 3 stimulation conditions. First, mock stimulation is used to characterize assay background levels. This dataset forms the basis for establishing the limit of detection and defining the transition between non-responders and responder status. Second, a combination of mock and FMC63 stimulation is used to establish the confirmatory cut point, which is especially useful when evaluating donors with elevated background responses. Finally, the distribution of responses obtained with the functional control is used to establish the minimal response criterion. This parameter serves as a sample quality control measure and helps prevent false negative interpretation. In many clinical studies, it is considered more informative than viability thresholds alone. I would strongly recommend having a biostatistician at this stage to ensure that the most appropriate statistical model is applied. In parallel with assay validation, we have also been developing positive control cells, specifically FMC63 prime T cells generated using custom peptide pools. PBMCs were isolated and cultured for 15 days in Tex-Mex medium, supplemented with human AB serum and the cytokines IL-15, IL-7, IL-12, and later IL-2. Peptide stimulation was performed on day 1 and 7, followed by a cytokine withdrawal phase designed to reduce background activity prior to analysis. For characterization, we used the CD19-CAR cellular immunogenicity flow analysis kit and observed specific responses against the 15-mer peptide pools. In addition, interferon gamma ELISpot analysis demonstrated positive responses against the 9-mer peptide pools. These findings have been reproduced across multiple donors and confirmed using independent statistical approaches. We are currently finalizing the priming process and moving into reagent qualification. Our goal is to include these cells as positive peptide-specific assay controls in future clinical sample runs. And with that, I would like to thank you very much for your attention.
Great. So in summary, during preclinical drug development, in silico epitope predictions and in vitro cellular immunogenicity assays are preferably used. For that, Peptivator peptide pools offer the most reproducible and reliable source for cellular immunogenicity assays, and standardized cell isolation improves reproducibility and reduces sample processing bias. At Miltenyi, we support immunogenicity complete workflows from primary cell isolation to functional immune cell assays. We have seen that systemic optimization of pre-analytical and analytical assay parameters enhances the reliable detection of even low-frequency cellular immune responses. And finally, as seen in the particular case shown here, optimized cellular immunogenicity assay performance was achieved through high-quality sample preparation and standardized antigen stimulation. Thank you all for participating. Once again, thank you to all the speakers, and see you next time.